This is Perry Marshall, and I'm here with David Quammen, and he is author of a wonderful new book called The Tangled Tree, a radically new history of life. And he's been a science writer for quite a long time. David, do I understand correctly? You wrote an entire issue of National Geographic?
I did. I wrote the May issue of National Geographic on the greater Yellowstone ecosystem. It was in observance of the 100th anniversary of the National Park Service, and they asked me to write the entire text for the issue. That was a very interesting privilege and responsibility.
Well, not bad. National Geographic is a very competitive little ecosystem in its own right. So,
yeah.
Well, that's great. And I became aware of your work through I had two of my clients actually send me links to your articles. One of them took a picture, and the other one sent me. And so it was the New York Times. You wrote a whole little excerpt of your book in the New York Times,
in Sunday Times. Yeah.
And then another guy wrote a piece in the Wall Street Journal. And I thought, well, all right, so what's going on here? And, there's been a quiet revolution in evolutionary biology going on, which I would say only in the last couple years really is starting to hit the public awareness. It's been going on already for a long time.
Yes, well, maybe several different revolutions.
Yeah. So in fact, I don't even want to put any more words to it. I'd like let's hear, you obviously figured out something was up. Yeah. And you took a very interesting angle to this, which we will explore. So, but why don't you give me, how would you describe the revolutions?
Right. Here's the revolution that I know about. Back in 2013, when I was sort of browsing for my next book project, I read something about the phenomenon called horizontal gene transfer. Horizontal gene transfer. Genes moving sideways across species boundaries. It's supposed to be impossible. Even across broader boundaries, bigger gaps. Genes moving from one kingdom of life to another. And my first reaction was, wait, no, that's impossible. Genes don't cross. I mean, when two closely related species hybridize, there might be some exchange of genes between two closely related species. Maybe they're only subspecies. But this was not that. This was something much more drastic. The idea that genes could move from one unrelated kind of creature into another sideways, horizontally, as opposed to vertically from parent to offspring, which is the classic way we think of genes passing down through time. Horizontal gene transfer. So I started reading more about it, came across some scientific articles by a fellow named Ford Doolittle, a microbiologist in Halifax, Nova Scotia, an American, but he spent his career in Nova Scotia. And he wrote a piece in the journal Science in 1999 about this that gathered together, it was a review article, and it made this subject sort of conspicuous within the scientific community. But I've written, previous books about Darwin and about evolution. I'd never heard of this stuff. And I read a little farther, and I came across this character, Carl Woese, who was the fellow who triggered this revolution back in 1977. A professor of microbiology, a little white-haired guy in Urbana, Illinois, at the University of Illinois. And he began this particular revolution that does not overturn 20th century Darwinian theory, but adds important elements to it, including the fact that we now know that incremental mutation is not the only source of innovation in a lineage upon which natural selection can work. We now know that there can be these big lumps of new genetic material that might come into a lineage, and I can talk about some of the specifics of that, but that can bring entire genes, entire clusters of genes, long stretches of DNA into a genome of a particular lineage, and then give some drastically new material, a major innovation upon which natural selection can then act. So that's the short version of this revolution that began with Carl Woese.
And this completely changes the speed of evolution too.
It can, in some cases, yeah. I mean, evolution can still be slow, but it can also be fast. For instance, antibiotic resistance among bacteria, and I talk about that in the book. You know, a single strain of bacteria might slowly evolve resistance to one antibiotic, say penicillin. Say, Staphylococcus evolves resistance to penicillin. But now we know that that gene for resistance to penicillin can pass instantaneously, not just from one Staphylococcus aureus bug into another, but from Staphylococcus into Salmonella, from Salmonella into E. coli. So that's fast evolution.
Well, and so that has reverberating consequences on the whole entire picture because, well, there was these classical arguments with Gould and different people about whether it was gradual or whether there were leaps, right? And punctuate equilibrium and all that. And so now we're changing the speed of evolution. We're totally changing the shape of the tree, which is what your book is about. And we're also, maybe you can get to this later, there's also all kinds of little tiny clues in the molecules as to the history.
Yes. That's why this field, I mean, what I've written is a history of a field and its consequences and our understanding of who we are, how we got here, and what the shape of the history of life is. I don't like to say it too often because it makes people's eyes glaze over, but this field is called molecular phylogenetics.
Right. Molecular history of body shapes.
When and I've been telling people, when you go, if you're going to write a book, when you go to make a pitch to your editor over lunch at a nice New York restaurant, when you tell him what your next book is going to be, do not tell him this is going to be a history of molecular phylogenetics. Do not say that. Maybe you want to say this is a book about the most important biologist of the 20th century that you've never heard of.
Well, I'm sure some people are probably bothered by the fact that you've basically written a biography of Carl Woese, that you've kind of wrapped this that way. I think it's great.
Well, people like to read about people. So when you write about science, in my view, you can't just explain science. You want to tell stories about the people who do science, the men and women like Carl Woese and Lynn Margulis and Barbara McClintock and Ford Doolittle who do the science and tell their stories to some extent. And then I try and weave them together into a tapestry that is fun to read as well as illuminating.
Well, I think his story really needs to be told. Now, I was familiar with him and his work for quite a while. I didn't know anywhere near what I learned reading your book, but really he was a very important person. So why don't you kind of tell the story of, so first off, he doesn't come from the usual sources of biologists for one thing, and he finds his way into this profession, and then he starts turning things upside down. So what happened?
Okay, so he's this guy. He goes to college at Amherst. He does a degree at Yale, a doctorate in biophysics at Yale. And then he goes to work for the General Electric Company in Schenectady, in the General Electric research labs. And he starts groping around, and he has no idea what General Electric wants from him. But he knows that he's interested in the deep history of life, deep questions. How did complex cells, like the cells that make up our bodies and all animals, plants, and fungi, fancy term, eukaryotic cells, meaning they have a cell nucleus and they have a fancy internal structure. How did those cells come to be? How did life begin? How did bacterial cells or whatever else was the first form of cellular life lead to these other things? So he's interested in those questions. He gets hired by the University of Illinois. He writes a book about the origins of the genetic code. Watson and Crick have just solved the structure of the DNA molecule, and they know that DNA codes for the arrangement of amino acids in proteins, like beads on a string. And the genetic code is a question of which letters in the DNA alphabet code for which amino acids. And people wanted to understand that. That was a puzzle that people were trying to solve. Francis Crick was trying to solve it, others. Woese comes along and says, I don't want to just solve that puzzle. I want to understand how the genetic code evolved. He always wanted to go a level deeper than other people. So he starts work, and he wants to know about the deep history of life. He starts using a very clumsy, simple, primitive method of genome sequencing to get fragments of certain genomic molecules in microbes. And he's using toxic chemicals and explosive chemicals and high voltage and radioactive phosphorus. He's got this mess, and he's got these wonderful assistants, men and women, who are helping him do this in his lab. And this is in the mid-70s, and nobody has fancy machines for sequencing genomes in the mid-70s. You got to do it by hand. This is hammer and nail sequencing. And so he gets fragments. He picks one molecule, one important, I call it his Rosetta Stone molecule, that's in the structure of all creatures, living creatures, bacteria, us, animals, plants, everything. One molecule that is basic to all forms of life. And he says, I'm going to sequence what I can of that molecule, fragments at least. And then I'm going to take those fragments and put them into paragraphs. And then I'm going to compare the paragraphs from different organisms. And he does that, and he discovers an entirely new kingdom of life, an entirely new form of life that nobody knew it existed. Creatures that look like bacteria through a microscope, but if you look at their genome, you look at the sequence of this particular molecule, they are more different from bacteria than they are from us, from complex creatures. And that group of organisms eventually become known as the archaea, because the archaea is an old archaeology, etc., because people thought maybe this is the oldest form of life. So Woese makes the front page of the New York Times, November 3rd, 1977. There he is, sitting with his feet up on his desk, wearing a pair of Adidas and a sports shirt. And here's this guy that nobody's ever heard of, Carl Woese, and suddenly he's the most famous, for 15 minutes, like Andy Warhol said, he's the most famous scientist in America. And then he disappears back into his lab and goes to work, follows up that discovery with other discoveries in terms of the shape of the tree of life. So that's a short history of Carl Woese, at least to the point when he became very influential with other scientists. Whoops, I lost your sound, Perry.
Are archaea familiar to regular people in any, is there any germ or infection or anything like that? Or is this just something that only biologists have any?
They are very little known to the general public for a couple of reasons. First of all, none of them cause infections in humans, as far as we know. Yeah, so they're not a problem. They're insignificant medically. You don't hear about them. You never go to a doctor and he says, oh, you got an archaea infection. Doesn't happen.
Okay.
So that's good. But also, at least at the beginning, these were all creatures that lived in extreme environments. They lived in high temperatures. They lived in hot springs in Yellowstone Park. They lived in acidic environments. They lived near thermal vents at the bottom of the ocean. They lived in oxygen-poor environments where they metabolized carbon dioxide and produced methane. They lived in extremely salty environments. So they were all these crazy extreme creatures that looked like bacteria but lived in places where bacteria generally couldn't live. And then Woese discovered that they weren't bacteria after all. Now we know they live in other places too, but that was the beginning. And that's part of why people are unaware of them.
Now, this also brought this whole concept of horizontal gene transfer. So can you explain what this actually is, first of all?
Okay, it is movement of DNA from one species into another. And sometimes that means it might be from across kingdom boundaries, across huge boundaries. It might be DNA from bacteria going into an insect.
Mm.
And that's supposed to be impossible. But it turns out it's not
Supposed to
impossible.
be.
Yeah, yeah. So the next question is, and if I can ask it of myself before you ask it, how? How does this happen? And
Hmm.
the short answer to that, there are complicated answers, but the short, simple answer to that is infection. The great
Mm.
Joshua Lederberg, a microbiologist in the 1950s, Nobel Prize winner, gave a name to this, coined the phrase infective heredity, horizontal gene transfers, infective heredity. He saw it in the 1950s among bacteria, from one kind of bacterium to another. But then in the when Woese's methodology took hold and then genome sequencing became much faster, better, and cheaper, people started finding this not just among bacteria, but among animals, plants, fungi, other kinds of creatures. Genes moving by infectious, and that infection, and generally that means a bacterium gets into a creature, gets into its reproductive cells, gets into the ovaries, the testes, the eggs, and the sperm, and infects those cells, the interior of those cells, intracellular parasitism within the cells, and then its DNA gets into the DNA of that particular reproductive cell, say in an animal. Or a virus infects somebody, and viruses can do this too by essentially what I simplified by calling it a drag and drop sort of mechanism. Virus can infect one creature, and it can pick up some of that creature's DNA in its own viral genome, and then the descendants, the replicants of that particular viral particle can infect some other kind of creature, and it can drop that DNA inside the cells of that creature. So there are three different mechanisms for this with fancy names, but the simple explanation is infection can carry genes sideways.
And isn't it true, it's not always just infection. Sometimes it's also receptivity. That at times cells are receptive to.
That's right, because sometimes what's brought in turns out to be helpful.
Mm-hmm.
That's again, the way evolution works. There are changes, there are innovations that natural selection works on, and usually if you change a genome either in a small way or a large way, you screw things up. You've thrown a monkey wrench into the works, and that lineage dies out. It's killed off by natural selection. But sometimes the innovation turns out to be good. It's serendipity. It's a happy accident, and that alien DNA that's come in is sometimes modified and repurposed. And I can tell you one really bizarre head-spinning case of this, so that it becomes advantageous. Okay, so 8% of the human genome is viral DNA. We know that now from genome sequencing. 8% of our genomes is DNA that has come down not by vertical linear descent through the entire animal lineage, but has come in sideways by infection over the last long period of time, 100 million years. Different events of infection. How can that get into our genome? Well, these are retroviruses. Retro, backwards. Retroviruses move backwards. They can put their DNA into the genome of a cell that they infect. Viruses, are not cells. It's worth saying that for people. Viruses are something else, but they infect cells. They get inside cells, and they use cell machinery to replicate themselves, and then the cell busts open and there are more viral particles and you get sick. With a retrovirus, like HIV, it inserts itself into the cell. It goes into the cell nucleus. It patches its own DNA recipe, its blueprint, into the genome of the cell. And then when that cell replicates itself, it replicates the virus, and eventually all those cells bust open and you've got a bloodstream full of HIV. Not good. We call that AIDS. But if it's a retrovirus, instead of infecting your immune cells, it infects your reproductive cells. It gets into the eggs, the sperm, the ovaries, or the testes. It patches itself into the genome. It becomes heritable. It gets passed down. Whether it's good, bad, or indifferent, it gets passed down. And that's how we've accumulated 8% of our genomes through viral retrovirus, captured retroviruses. Now, Some of those are dysfunctional. It's just DNA that's there, what scientists used to call junk DNA, thinking that it didn't do anything. They don't say that anymore because they realize it's much more complicated than that. It's just, DNA that might or might not have a function, but we don't know the function. But one of these viral genes that's come into the human genome, which is called, it's got a fancy name, syncytin-2. Syncytin-2. Mm-hmm. Syncytin. Discovered by a guy named Thierry Heidemann in Paris and his group. It discovered to be a useful gene. It's come from a virus. It's gotten into the human genome. Originally, it formed sort of an envelope membrane around virus particles, and now it forms a different kind of membrane in humans. It forms a membrane between the placenta and the fetus in a pregnant woman. And that membrane mediates between the placenta and the fetus in a completely crucial way, bringing nutrients into the fetus, carrying waste products out of the fetus, possibly also defending the fetus from the mother's immune system, because the fetus has a genome that's only 50% the same as the mother's. Mm-hmm. This membrane, which is completely necessary for successful pregnancy in humans, comes from a viral gene that was captured sideways sometime in the course of mammalian evolution.
So in your book, Tangled Tree, there was a little detail about that I just thought was really fascinating. I had never picked up anywhere else. And it was that that mammal, not just mammals, but maybe creatures similar to mammals, like kangaroos are related to mammals, but they're not exactly right.
Kangaroos are mammals, but they're not placental mammals.
Oh, okay.
Yeah.
But there were multiple places in the family of higher animals that had co-opted different versions of a virus to make a placenta. So okay, now I thought, oh my goodness, that would be an example of convergence. I don't know if you can talk about that, but that this had been engineered 4 different times, 4 different ways.
Yes, that's right. That's what Thierry Heidemann's work has shown, and that's what I describe in the book, that this syncytin-2, it's not the only gene that does this. There have been a series of versions. Some of them are still in our genome. For instance, when the first animal that was laying eggs, a reptile or a bird, started evolving into a mammal with internal pregnancy, how did it do that? Well, it had to have some way of starting to form a placenta and intermediate between the placenta and the fetus. And one of these genes was probably captured from a retrovirus at that point, and it was put to this use. But then, maybe 10 million years later, another individual pre-mammal got infected with another virus that entered the genome, and it was an improved version, and it was adapted. So this has happened a series of times, and it can be seen in the mammal genome. Different mammals have different versions of this gene. All mammals have to have some version of this gene. So it had to come in a primitive form originally once. But since then, it has been improved upon, and it's been improved upon not just by sort of mutation and slow evolution of the gene, but by capturing of more viral envelope genes and putting them to use in improved ways. That was amazing to me. I flew to Paris to see this guy, Thierry Heidemann. I emailed him when I started reading his book, or started reading his papers, and I said, whoa, what? This is amazing. I emailed him, very nice man. I said, if I come from Bozeman, Montana to Paris, will you talk to me for 1 hour about your book? And he said, yes, of course, sure I will. So I flew to Paris. I'm not rolling in money, but I knew this was going to be worth it. I flew to Paris. He's such a nice man. He says, oh, you're staying at such and such hotel that's near my apartment. I'll pick you up. We'll drive across the city of Paris, 45 minutes, the world's most elegant scenic commute to my institute So we did that. We drove across the city and we kept talking. I talked to him for 7 hours that day about this work, and it was worth it. And then the next day I got on a plane and came back to Montana.
Wow. That's fantastic. Well, so the Wall Street Journal guy was a little grumpy, and he was like, hold on, everybody. Let's just remember that natural selection and Darwinian evolution is still in place. Like, you know, like he seemed kind of rattled. Now, I think I completely understand why he's rattled, but why is this a revolution? And why do you have to start thinking differently about biology? And why do people feel threatened by this?
Okay. Well, first of all, I'll answer that. First of all, I want to say that reviewer is an evolutionary biologist and writer named David Barish. I've known of his work for a long time. I don't like to complain about reviewers. Everybody has their point of view. I reviewed too. So there were things that he didn't quite like, and that's fine. He's in touch with that. That's great. Thanks for the attention to the book.
Right.
Secondly,
all publicity is good publicity, right?
Yeah, right. Secondly, how is this a revolution? Well, people are very defensive of Darwin. Darwin was a great scientist. I've written a biography of Darwin. I love Darwin. But Darwin published The Origin of Species in 1859, knew nothing about genetics, knew very little about microbiology. He did amazing work, and he has remained right on most things for 150 years. But Darwin was the Isaac Newton of evolution, and then comes the Einstein of evolution, who modifies Newton without falsifying Newton, and that's the analogy I use. And I think Carl Woese and the work that followed him, that's the Einsteinian revolution in understanding better what Darwin so foresightfully sketched out. And the modifications are, and we talked about some of this at the beginning. The biggest modification is in terms of the pace and the origin of variation, of innovation in genomes. Natural selection, I believe that natural selection, even Darwin didn't believe it was the only mechanism. There's also genetic drift, which is sort of a random process. It happens among small populations on islands in particular. We could talk about that if you want, but the real headline news of this second half of the 20th century revision of orthodox Darwinism is about the pace and the origin of the innovations that get into a gene upon which natural selection works. And the biggest of those changes is the awareness that horizontal gene transfer can be bringing whole chunks of new genes and fragments of genes into a genome, and then natural selection has to sort that out.
Well, when Einstein came out with his theories and quantum mechanics and everything, there was a whole cadre of people. They didn't like it. They didn't want it.
Right.
Like, don't bring that around here. This is all going to be proven wrong. I believe there was some kind of book that came out in the 1920s or something called 50 Scientists Against Einstein or something like that. It was like, you know, we have all signed our name and this is wrong and we are going to squash this heresy.
Yeah.
You know, so, but it shows you that evolution isn't just random and accidental. It's not just gradual. It means there's immensely complex things going on that we barely understand. And some people kind of like to tell everybody that we have it all figured out, and like, not even close.
Yeah.
So
that's one of the things that makes evolutionary biology such an exciting field, is that we're still figuring things out, still figuring important things out.
Well, I think that you could make a very compelling case for making Carl Woese the central modern figure in evolution. You could also make a case around Lynn Margulis. You could also make a case around Barbara McClintock. I thought it was very interesting, your story of 2012, like, or 13, like, hey, how come I haven't heard anything about that? I had a very similar moment in 2006 when I discovered Barbara McClintock. And I had been reading evolution stuff for a good 2 years, maybe more. I'm like, I've never heard about this.
Yeah.
And she discovered that a plant, if she hacked a plant's genome, that plant would actively restructure its genome in real time, repair the damage, and make an evolutionary change right there on the spot. No, which completely conceptually changes the landscape of evolution all by itself. Yeah. Not to mention what Woese came up with, not to mention what Margulis. I didn't even find out about Margulis till a couple years later. And so it was like there was the standard textbook version of evolution that was shamefully out of date. I mean, yeah. It's not like this happened yesterday.
Yeah. Well, and I don't know. I mean, I talk about Barbara McClintock in this book. I don't know the particulars of the particular experiment you're talking about, but she was important to this particular narrative connection of ideas because she discovered in corn, which is the plant that she worked on, maize, corn, she discovered these things that we now call transposons, jumping genes, that genes were jumping around within the genome of corn, from one spot on one chromosome to another spot, jumping around. And she called them control elements, but later they became known as transposons. And that's what she won the Nobel Prize for in, like 40 years after she did the work in 1981 or something like that.
Right.
And then other people, and this is why McClintock is in my book, other people discovered that these transposons didn't just jump around within the genome of one creature, but they could jump from creature to creature, species to species, again by infective heredity. There's a fellow named Cedric Fichotte, now he's at Cornell University, who's done some really interesting work on this, he and his group. And I went and interviewed him, and he's a character in the book too. These transposons jumping from one species into another. I was thinking about, I can't remember if it was talking earlier with you or somebody else about the classic story of the peppered moth in England. You know that story?
That wasn't me.
Oh, okay.
I know the story, but I don't know that my listeners necessarily know that story.
Should we go there?
Yeah, sure.
Okay, so there's this moth, this white moth with little dark flecks on it, that's living in England in the 19th century. It's been there forever. It's white with little flecks. And then the Industrial Revolution happens, and this moth is used to roosting on the side of tree trunks that are white, like a birch tree. But then smoke from the mills of Manchester turn all the tree trunks black, and suddenly these moths are vulnerable to birds that can come and see the white moth on the black trunk. So within a fairly short period of time, the moths turn black. They evolve into a melanistic, a black form, and now they have protective coloration again. It's one of the classic stories of classic Darwinian evolutionary theory. And people who have gone back and looked at the research, looked at this particular moth, and there was one point when they thought that, well, this story just didn't hold up. And now the latest thinking is that it does hold up. But what they found recently, I just read this in a book that I'm reviewing for the New York Review of Books. I found mention of a study that shows that change from the white version of the moth to the black version didn't happen by incremental mutation, little by little by little by little, over the course of 20 or 30 years and 40 or 60 generations. It happened like that because a transposon, 22,000 DNA letters long, got into the genome of that peppered moth and changed it instantly from white to black. A transposon, horizontal gene transfer.
I did not know that story. Now, speaking as an engineer and being pretty familiar with McClintock's work, my interpretation of that is that the physiology of the moth got a piece of genetic material and in some sense knew what to do with it. We know that when germs are under stress, they'll hypermutate. Like they'll take certain parts of their genomes and they'll start like, well, let's try this, and they'll threatened by antibiotic, it'll go looking for genomes that it's like, hey, what do you got? What do you got?
Yeah, right.
Right.
Yeah. One of the first. Go ahead.
No, go ahead.
Oh,
Sorry.
I was going to say one of the first mutations sometimes in a major evolutionary change is a mutation that increases the mutation rate of that genome, just makes it worse at copying itself precisely, and that mutation can lead in myriad directions, but in two extreme directions. One is toward good and one is toward bad. If suddenly you have a higher mutation rate, you copy your genome and it keeps getting different and it keeps getting screwed up. Or you have a higher mutation rate because you're under stress, you're in an emergency situation, and you need something new. You need a new tool. So mutation, bingo, suddenly a new tool appears. Now, you may disagree with me on this, but I would say that appears randomly because you've got a high mutation rate, and then natural selection seizes it.
Well, as best I can tell, the high mutation rates happen in hot spots. And so like here's an example that I'm somewhat familiar with is you'll have hypermutation of one in like there's a mechanism called VDJ recombination, which is an immune system thing. And the cell will hold most of the genome constant while rotating values in a very certain small part. And it's sort of like, it's like, well, I want a door and I need a doorknob, but is it a white door? Is it an orange door? Is it a blue door? Is it a yellow door? And it's a permutation.
Or I need something at the end of my leg, and I've tried a wheel and I've tried a flipper. They don't work in my environment. I need a foot. Yes.
And I would just, my personal perspective from all my research is that the exact predictive nature of such mutations is very poorly understood. And I mean, there's a lot of room for research there. And I would hypothesize, no, that's not strong enough of a word. I would insist that to simply say that it's random slows the process of finding the systematic mechanisms that are underneath that. And
okay, I think so to say, I mean, they talk about the null hypothesis, and so which is essentially the default hypothesis. And you're saying to say that the default hypothesis is randomness will not lead you as quickly to new insights as to open yourself to the possibility that it's not random.
Yes.
Okay. Well, I think that's a fair position. I don't have a quarrel with that at all. Do you know Richard Lenski's work and his experiment?
Yes, I do.
I was looking at that again today. Should we talk about that at all? That seems to me really.
Yeah, I met him at a TED conference in 2010, and we talked about this. So go right ahead, and may have a little serendipity here.
I'll sketch the experiment, and you tell me what he said to you. I've met him a few times, but I've never really gotten into this with him. So here's a guy, an evolutionary microbiologist at Michigan State University, Richard Lenski. He's probably about 63 years old now. And 30 years ago, he decided to conduct what he calls a long-term evolutionary experiment, LTEE, long-term evolutionary experiment. So he took 12 different populations, genetically uniform populations of E. coli, the bacterium, the common gut bacterium, and he separated them and he started them, giving them nutrients and keeping them separate, and let them evolve. And then I think each day he would take a sample from each of those and put it into another dish or another test tube, so that he was tracking how they changed just in this environment of glucose and citrate and some other chemicals. And it's been going on for 30 years. He's got more than 65,000 generations of evolution from these 12 original bacterial strains. And he's been doing this, and he's been sequencing genomes to see how do they change? What's the rate of mutation, and what are the results of mutation, and how is natural selection working in these things? And he's found some very interesting results. For instance, one of his strains evolves the ability to live on citrate instead of on glucose. It generated an entirely new gene. It was like, if humans developed a gene where we could, like termites, we could live on cellulose, we could live by chewing trees. That's what these microbes did. So that's part of what I know about Lenski. But what did you and Richard talk about?
Well, so I'm familiar. So it's 60,000 generations of bacteria, and what happens here. Well, so I've never talked about this in public, but I
great, do it now.
So we're talking about it now. So I read one of his papers, and he said that this mutation that made citrate, so it could digest citrate, and it couldn't before, it had happened 2 or 3 times separately, and he said in his paper that it happened randomly. And I said to him, I said, the data from your own paper does not support the idea that this happened randomly. I said, the statistical chances of that same thing happening 3 times in 1 million base pairs is way too small. I'm an engineer, I know probabilities. I said, that was some kind of systematic mechanism that was going on. And I said, I actually have a question. I said, I was digging around in some old journals, and I found this debate between you and James Shapiro in the 90s. And you were saying that evolutionary mutations are random. And Shapiro was saying, no, it's natural genetic engineering. It's the kind of stuff that McClintock and Margulis were coming up with. And I said, Richard, what would be your perspective on that debate that you were having 20 years ago with Shapiro? Shapiro is a geneticist at the University of Chicago, and he was a protege of Barbara McClintock. And he said, Shapiro's, I'm not quoting him exactly, but sort of like, Shapiro turned out to be correct. And I said to him, I think that there are predictive mechanisms in the mutation machinery that are looking for things that will work. Mm-hmm. I said, we don't know how predictive they are. That's a complete mystery at this point. But I would go looking for a predictive mechanism where, in some sense, the bacterium knows that it's trying to accomplish something. Now, I have a couple comments about his experiment. I think his experiment is inherently incapable of producing large evolutionary changes, because think about it. There's no opportunity for massive viral infections. There's no opportunity for massive horizontal gene transfer events from wildly different organisms.
That's right, yeah.
There's no opportunity for symbiotic mergers either. Okay,
and Margulis pointed to as a hugely important factor.
Right, and so he's got these isolated strains of bacteria, and they're just so. here would be my prediction. They could run that thing for 200 years, and what they'll basically get is dogs will always breed as dogs. If you want something radically different, you have to do something. So Quan Zhang at the University of Tennessee, he's a friend of mine. He's a friend of mine because I hunted him down, kind of like Theory in France. Like, I was like, how come this guy didn't get a Nobel Prize? Like, and well, here's what Quan Zhang did. He put Amoeba proteus and X bacteria together, and basically they fought like cats and dogs. Okay, but at the end of 18 months, he actually got a symbiotic merger where the bacteria and the amoeba had gone inside the amoeba. Both of them had shed DNA that they didn't need, and they had consolidated functions like a corporate acquisition, where we don't need 2 accounting departments and we don't need 2 HR departments. Okay. And they had.
I got to read this paper. I'm going to make a note about it.
Maybe after we talk, I'll send it to you.
Okay.
I had to dig hard to find this thing.
That's
so
very interesting, because that's like an experimental replication of the historical endosymbiosis events that Lynn Margulis wrote about all her career, right?
Yes.
Yes. Recreation of that. Well, that's
yes.
That's pretty interesting.
And so by the time it was done, if he separated them, they would both die. And that's inherent to the proper definition of a symbiotic merger, which is they have become so interdependent. That's like, well, like you take them apart, like there's no accounting department and this one has no HR department, and like, right?
That's what they call them, obligate symbionts, because they're obligated to have each other, otherwise they die. Yeah, that's very interesting.
And the conflict in like that, like most of them died, and like it was kind of ugly. I'm in business strategy. I can tell you that most corporate acquisitions don't work out either. Like, we've all heard stories about Daimler Chrysler. Like, yeah, that was a good idea, right?
Ted Turner is still shaking his head about how his merger with Time Warner went wrong.
So I think that that major evolutionary events only come from large imports of foreign material, which by definition are not likely to work out well most of the time. Well,
whether you're right or not, or partly right, that argument is plausible, and there's a lot in my book that will help you make that argument. And I'm not sure that I can sign on to the word only, but
sure.
This stuff as a source of innovation has huge implications, and that's why I wrote the book, and that's why I hope people will read it. It has huge implications for understanding what may have been and may continue to be some of the really important events in evolutionary history, such as the ability of animals to have internal pregnancy. That's a pretty important innovation.
Right.
Maybe
could you just connect the dots a little bit about what you just said, because that was a really important point. Okay. And then I want to circle back to Carl Woese.
Okay, this is back to those captured viruses and Thierry Heidmann's work, and the viruses that get into our genome and make possible this membrane between the placenta and the fetus. So infection by those viruses and acquisition of those genes that were modified and repurposed in humans, instead of making a viral envelope, they make this membrane, made possible the development of internal pregnancy with a placenta and a fetus held inside the body, so that the female didn't have to lay eggs, set them down on the ground with a calcium shell around them, and then protect them. Instead, she had the option of running away and taking her fetus with her. She didn't have to leave it on the ground and then try and protect it. That was an important, we still have birds, so as Thierry Heidmann said to me, birds still exist, so it's not a terrible way to do it, but there are other ways that have, in some ways, better opportunities. Maybe mammals became dominant on this planet. Maybe we became the dominant mammal, partly because of that viral infection that allowed placental pregnancy.
Well, there was another little detail that I thought was interesting, that the coding sequences that came from those viruses were related to the virus's ability to suppress the immune system of the creature that it was invading.
Right.
And then the placenta performs an immune suppression function, right? And so it does the same thing in a different context, and then it's been used 4 different times independently,
right.
Which I just think it's fascinating.
And Thierry Heidemann made the point to me. He said, this is our hypothesis, that perhaps the first utility of this viral gene in animal lineages that became mammal lineages, possibly the first utility was this immune suppression. Because if you've got a fetus you're holding internally and not an egg you're laying on the ground, and that fetus has the father's genes in it as well as the mother's immune system is going to want to get rid of it, because those father's genes are alien material. So you have to protect it, and that's one of the functions that this membrane does. Thierry Heidemann said to me, we can't prove it, but we hypothesize that perhaps immune suppression was the original purpose.
Got it. Very interesting. Well, let's land the plane here. Why don't you kind of circle back to Carl Woese and talk about, you talked about what he discovered, but we haven't really talked about how it impacted the profession, how he's come to be viewed, and why he should be much more central, in your opinion, than he has been.
So I mentioned his primitive method of sequencing genomes before we had fancy machines. He did one very important thing, and it was probably more important than discovering this whole new branch of life, this kingdom of life called the archaea. He chose a particular molecule as the Rosetta Stone for the history of life. And it's a molecule, it's an RNA, not a DNA molecule, and it's an RNA molecule, single-stranded rather than double-stranded. And it performs a structural function in the organ that all cells contain that changes DNA into proteins and changes information, genetic information, into physical bodies. They call it the translation apparatus. So this all living creatures need a translation apparatus, and this particular RNA molecule is there in all of them. So he said, I'm going to look at that. That's going to allow me to compare all forms of life to one another and draw a tree of life, a new tree of life. So in 1990, he proposed a new tree of life. But then other people picked up on his method and started comparing other creatures to one another, and genome sequencing became faster and cheaper and easier. So it wasn't just fragments anymore, it was whole genomes. It wasn't just this Rosetta Stone molecule, it was the entire genome of creatures, and they started finding horizontal gene transfer. And they confirmed Lynn Margulis's theory of endosymbiosis. We didn't talk much about that, but the fact that we have bacteria inside our cells that have been turned into internal organs. They confirmed all this with genome sequencing, and they said, no, your tree of life, Woese, which is just, different branches coming out, that's not correct, because we got a lot of branches that are converging. You were the first one who used the word converging. Converging is a really important term here. Branches converging, limbs growing sideways into one another, carrying information from branches carrying information from one major limb into another. That's why my book is titled The Tangled Tree. And that's how Carl Woese's early work influenced a lot of other scientists who redrew the tree of life into this tangled shape that we now know is the history of life on Earth. Carl Woese, he was bitter at the end of his life. He came to hate Darwin. He came to think that he was a more a greater scientist than Darwin, which was not, I believe, not true, but he was a very great scientist. He never won a Nobel Prize. McClintock won a Nobel Prize for biology. There's no biology category. There's only physiology and medicine. She won in physiology and medicine for doing biology. Watson and Crick won in physiology and medicine for doing biology. Woese hoped that he would win one. Never did. And therefore, and then he died in 2012. He was well recognized. He had a MacArthur Genius Fellowship. He was a member of the National Academy of Sciences. He got a lot of awards in his life, but he never regained that 15 minutes of Andy Warhol fame that he had on the front page of the New York Times, November 3rd, 1977. So when he died, scientists all over the world said, "Oh my God, the great Carl Woese has died," and everybody else said, "Who?"
Right.
So I'm trying to answer that question, the who, with this book. I'm trying to put him back on the map.
I think it's absolutely proper. Like, if somebody said, just in my personal opinion, is the Tangled Tree like, you know, the definitive, like, oh, you know, work about evolution? Well, no. What it is it's bringing a very important figure to light. I would say I would look at it like this, personally. I think Darwin was remarkably correct given how incredibly crude his methods were. I also think that the modern synthesis which followed him was a step backward, not a step forward. There was a lot of wrong assumptions. Like, for example, Darwin embraced Lamarck's theories, which turned out to be right only in the last 20 years, really, you know.
Yeah. Well, I mean, the inheritance of acquired characteristics. That particular aspect of Lamarck, right. People keep trying to kill that, and it's something that is very hard to kill, that idea. And well, you're right. Horizontal gene transfer, you could say that that's the inheritance of acquired characteristic. Yeah. So you're right. And Darwin did embrace it. He called it use and disuse. It's right there in The Origin of Species.
And so now I think in the landscape of the contributions that people have made, in my personal opinion, I think Darwin is overrated. I think if you want a comprehensive understanding of what's really going on, okay, well, yeah, we already know about the Darwin stuff. Most people, they don't understand what Carl Woese did. They don't understand what Margulis did. They don't understand what McClintock did. And those, like, if you take those three people and you put them together, you get a radically, it's like we got a supercharger, we've got a 10-gear transmission, we've got a cruise control, we've got autopilot. Like, we've taken a very crude vehicle and made it much more sophisticated and interesting. And look, if you don't understand retroviruses, horizontal gene transfer, and transposons, it's going to impair medical research, disease treatment, all this genetic engineering. I mean, these are, and given how we can now edit genes as easy as Microsoft Word with CRISPR, like, we have to appreciate these things, and we have to appreciate the complexity of what we're messing with, because this did not come together by a random accumulation of point mutations. I mean, it's just right. So there's a certain amount of reverence that we need to bring to the table.
Yes, I agree, and I think people should be glad that evolutionary biology is not a finished science. It's an unfolding science, folks. Things are happening yesterday and tomorrow that are really exciting and important. New ideas, new insights. This is a bubbling science. Well,
so run out and buy this book. It's probably at Barnes and Noble or Amazon or wherever you like to buy books from. The Tangled Tree: Radical New History of Life by David Quammen. People quibble about the word radical. I say it is. Compared to what is in most, if you go to Barnes and Noble and you go to the evolution shelf, most of the books there are out of date. Jerry Coyne, out of date. Richard Dawkins, out of date. Charles Darwin, you know, God bless him, but out of date. This is up to speed, and
you heard it here from Perry. You trust Perry. You heard it from Perry.
Well, look, it's
Thank
true.
You,
I mean, look, I think that what Carl Woese, Lynn Margulis, Barbara McClintock, and similar Doolittle, similar type people, the dynamic evolutionary discoveries that they made are the biggest untold story in the history of science. And you took a very private, sort of crotchety guy who's really brilliant, and you always find that these geniuses they're eccentric and interesting, and they're into weird things like jazz or whatever, you know, and you dig out these stories like, wow, this is really interesting, and I appreciate what you've done here. I think people need to understand this, and we got to get evolutionary biology out of the 19th and 20th centuries. It's 2018, baby. You know, like the world is changing, and so yeah, good job, great. I also want to compliment you on the storytelling. Having written an evolution book myself, it is really hard to take cold, hard scientific facts and wrap bacon around them, but you did it. Okay.
Thank you.
There's a lot of bacon in the kale salad. Okay.
I love hearing that. Thank you, Perry.
You need to get some appreciation for that. So thank you. Good job.
Great talking with you. I really enjoyed it.